Hello, my name is David MacTaggart and I'm from the Department of Plant Sciences at the University of Saskatchewan. Today I'll be sharing my research regarding estimating the plant characteristics of cicer milkvetch using a remote sensing approach. After my presentation here today, I hope to have addressed the following key questions. Why do forage breeders care about cicer milkvetch? What are the potential benefits of using remote sensing in forage breeding? What are the objectives of this research? What were the methods that were used to answer those objectives? What are the results from the 2020 field season? And what conclusions can be drawn from those results? And how can these conclusions inform future research? So why do forage breeders care about cicer milkvetch? As a non-bloat perennial forage legume, cicer milkvetch provides consistent and nutritious forage in long-term pastures within the moist areas of the Canadian prairies. Research has shown that it is compatible in mixtures with bunch-type grasses or grasses with a short rhizome so that the cicer plants can establish in the early years after seeding. In summary, the persistent yield, late-season quality, and compatibility in mixtures makes cicer milkvetch a desirable perennial forage for stockpile grazing systems, where a pasture is harvested early during the growing season and then left to stockpile until the fall or early winter when it is grazed a second time. This is a target that forage breeders can work towards. The potential benefits of remote sensing for forage breeding programs can be divided into three general categories. The first is time savings. Remote sensing via unmanned aerial vehicles or UAVs can screen large areas in a relatively short amount of time, which could offer breeding programs the chance to save on labor costs, while also potentially screening larger breeding populations. Shifting from a qualitative rating scale to a quantitative measurement. And this has been attempted in crops like perennial ryegrass and white clover to go from a manual rating of something like green area and shift to where remote sensing is used to count the number of green pixels in a plot. And finally, a non-destructive measurement of yield. In lentil, remote sensing was used to estimate canopy biomass by measuring the volume of that canopy. This trait canopy biomass could be even more valuable for forage crops, for which that biomass is the primary unit of yield. This component of my master's research can be divided into two major objectives, which are first to compare a thresholded NDVI to a blue NDVI approach for estimating the dry forage yield of cicer milkvetch, and second, to investigate the potential of an estimate for canopy volume to measure the dry forage yield of cicer milkvetch. To clarify some terms that I've been using so far, NDVI refers to the Normalized Difference Vegetation Index, which is a normalized relationship between near-infrared and red light. And then blue NDVI is that same normalized relationship, but instead of red light, we swap in blue light instead. And these vegetative indices were used to develop our drone traits, one of those being a green area estimate and another being a canopy volume estimate. This study is based on a cicer milkvetch nursery located at the Livestock and Forage Center of Excellence, south of Quebec, Saskatchewan. 27 populations of cicer were examined for this study that were placed in a nursery that was arranged in a completely randomized design with 2 to 3 reps per population. There were 2 harvest dates per growing season to simulate a stockpile grazing system. The first harvest took place in late June and early July, and this was the summer harvest. Then the plants were allowed to stockpile until the second or stockpile harvest in mid-October. Our remote sensing pipeline can roughly be divided across these major sections. Flight planning was conducted in drone deploy, so that then we could collect our data using our MicaSense Red Edge MX sensor mounted on our DJI Matrice 200 V2 drone that we flew at 15 meters of altitude above our plots. Image calibration was then conducted in Pix4D, and our image processing entailed making the point cloud, orthomosaic, and the digital surface model. For our plot segmentation, we conducted this in QGIS on a per replication basis. Reflectance extraction entailed first setting a threshold for both blue NDVI and NDVI to act as a soil mask. I chose the threshold after visual inspection of the nursery using these two vegetative indices, whereby the most soil could be excluded and the most vegetation could be included. The image-based plant area was based on the sum of all of the pixels deemed vegetation. Canopy volume was the product of the green area of each of these pixel counts and the mean height of the canopy in the replication based on the digital surface model. Statistical analysis was conducted in R. The models were primarily mixed models with replication as a random effect. And while there were violations to the assumption of equal variance of the model residuals in some cases, this is generally considered to be acceptable in forage breeding as yield seldom falls under a normal distribution. So now we can dive into the results addressing that first objective, which was to compare a thresholded NDVI to that of blue NDVI for estimating the forage yield of cicer milkvetch. This first objective can be divided into two sections based on the harvest dates during the growing season. Right now, we're looking at data from the summer harvest in 2020. Based purely off of the correlation coefficients, the remote sensing based green area estimate using blue NDVI or BNDVI is more highly correlated to the dry forage yield of cicer milkvetch when we compare it to NDVI. And this makes sense when we look at the scattering patterns, where NDVI demonstrates a flaring pattern that we're seeing on the left, whereas with blue NDVI, those data points cluster more closely to the trend line. The low correlation of NDVI-based green area could be partially explained by the light quality factors I'll explain later. And it's important to note that in these plots, there was an outlier that was removed because it had a forage yield that was far greater than the rest of the replications. Now switching to the correlation coefficients from the stockpile harvest in 2020. In similar aspects to that summer harvest, we see that the rank of these two green area estimates are the same. BNDVI-based green area continues to outperform NDVI to estimate that yield of cicer milkvetch. However, in contrast to the summer harvest, the difference between these two remote sensing strategies is much less, and we see that those data points for NDVI have contracted to be closer to the trend line. Now, so the main conclusion that we can draw from here is that a BNDVI-based green area strategy outperforms that of NDVI to estimate the yield of cicer milkvetch at both the summer and stockpile harvest. So now let's switch to address that second objective, which looks at the potential of an estimate of canopy volume to measure the dry forage yield of cicer milkvetch. What we're looking at here is the summer harvest on the left and the stockpile harvest on the right. In both cases, canopy volume is based on the BNDVI green area multiplied by the mean canopy volume across the replication for the drone's digital surface model. And in both cases, we see that this mean canopy volume delivered a highly significant correlation with the forage yield of cicer milkvetch. However, similarly to what we saw with simply the BNDVI green area, the highest correlations with yield were seen during the summer harvest. So now that we've reviewed our results pertaining to our two objectives, what conclusions can we draw from this 2020 data? First conclusion, a green area estimate based on BNDVI was more effective at estimating the yield of cicer milkvetch compared to NDVI. NDVI had a few issues in its ability to detect the vegetation of cicer milkvetch. At the summer harvest, as seen in that picture there, because of the lighting conditions on the day of the flight, the soil had a greenish appearance to it, leading to that large blob in the bottom left corner of the summer harvest picture. At the stockpile harvest, the stems of cicer milkvetch become white as the plant matures, which is a color that NDVI tends to classify as soil instead of vegetation. Second conclusion, generally we saw that a BNDVI green area estimate was more effective at tracking the forage yield of cicer milkvetch when compared to our derived plant volume estimate. And this could be because cicer milkvetch grows in a prostrate manner. There wasn't a great deal of variation in the height between populations. This mesh trick might be more effective for meadow brome grass, which is the other species included in my project. And then finally, the third conclusion, the highest correlations with forage yield came from the summer harvest. And so a future question to ask might be, is this partially explained by the reflectance of these plants hitting a plateau as they progress towards the stockpiling harvest date? Here is the past research I used in developing my presentation, which concludes what I have for today, and I'd be happy to answer any questions from the audience.